Hello students, welcome to readmedprep Academy channel. Today in Applications of Biotechnology Part 2, we are going to talk about the gene therapy, stem cell therapy and stem cell banks. First let us move on to the gene therapy. If a person is born with a hereditary disease, can a corrective therapy be given for such disease? Yes, this can be done by a process known as gene therapy. This process involves the transfer of a normal gene into a person's cells that carries one or more mutant alleles. Expression of normal gene in the person results in a functional gene product whose action produces a normal phenotype. Delivery of the normal gene is accomplished by using a vector. The main thrust of gene therapy has been directed at correcting single gene mutations, as in cystic fibrosis and hemophilia. In this image, you can see the gene therapy that can be given in cystic fibrosis. The genetic material is taken, that may be in the form of a DNA, RNA or oligonucleotides. They are packaged into a therapeutic vector. The therapeutic vectors may be viral, adeno-associated virus or lentivirus, or non-viral like the exosomes, liposomes and lipid nanoparticles. And these can be directly given into the individual for therapy, or cell-based delivery can also be done, where the cells are collected from the lungs and the CFTR correction is done, in which the addition and editing is done, and this is again expansion of the corrected regenerative cells is done, and the corrected cells are transplanted to the lung for therapy. At present, most genetic diseases have no effective treatment, and so gene therapy could offer hope for many people. There are two strategies involved in gene therapy. One is called the gene augmentation therapy, the other one is called the gene inhibition therapy. In gene augmentation therapy involves insertion of the DNA into the genome to replace the missing gene product. And gene inhibition therapy, which involves insertion of the antisense gene, which inhibits the expression of the dominant gene. Here in this image you can see the gene augmentation therapy fixing the genes that are not working properly. Example, cystic fibrosis. In the image you can see where the cell with the non-functioning gene is taken and the functioning gene is introduced into that cell so that the cells start functioning normally. In gene inhibition therapy, where the harmful genes are blocked, example, some cancers. You can see the cell is taken containing the faulty gene and the genes are introduced which blocks the faulty genes, and the new gene product blocks the faulty genes and the cells start functioning normally. The two approaches to achieve gene therapy are somatic cell gene therapy and germline gene therapy. Somatic cell gene therapy involves the insertion of a fully functional and expressible gene into a target somatic cell to correct a genetic disease permanently. Germline gene therapy involves the introduction of DNA into the germ cells, which is passed on to the successive generations. Here you can see on the left side the germline gene therapy, where gene therapy is given to the embryo. And this gene therapy, all the cells are targeted, including the germline cells. On the right side you can see the somatic gene therapy, where the embryo is not given any gene therapy and the entire human being is given the gene therapy, and all the somatic cells are targeted and there is no involvement of the germline cells. What are the differences between the somatic cell gene therapy and germline gene therapy? In somatic cell gene therapy, therapeutic genes are transferred into the somatic cells. In germline gene therapy, therapeutic genes are transferred into the germ cells. In somatic cell gene therapy, introduction of the genes into the bone marrow cells, blood cells, skin cells, etc. Here in germline gene therapy, genes introduced into the egg and the sperms. In somatic cell gene therapy, will not be inherited in later generations, whereas in germline gene therapy it is heritable and passed on to the later generations. Gene therapy involves isolation of a specific gene and making its copies and inserting them into the target cells to make the desired proteins. It is absolutely essential for gene therapists to ensure that the gene is harmless to the patient and it is appropriately expressed and that the body's immune system does not react to the foreign proteins produced by the new genes. The first clinical gene therapy was given in 1990 by William French Anderson, who is called the father of gene therapy, for a 4-year-old girl with adenosine deaminase deficiency, otherwise called ADA deficiency. ADA deficiency or SCID, which is shortly called and expanded as severe combined immunodeficiency, is an autosomal recessive metabolic disorder. What are the cause of SCID? It's a chromosomal 20 linked SCID where the mutated ADA is present in 25% of all cases and mutated gamma C receptor for IL-7 cytokine X-linked SCID. In 70% cases are other causes which are not monogenic. Adenosine deaminase, what is it? It's a glycoprotein and acts as a hydrolase, catalyzing the deamination of adenosine into inosine. Here you can see the reaction. Adenosine plus water gives inosine plus ammonia. Adenosine is toxic for B and T cells. ADA is essential for the proper growth and function of the infection-fighting T and B lymphocytes. It is caused by the deletion or dysfunction of the gene coding for ADA enzyme. In these patients, the non-functioning T lymphocytes cannot elicit an immune response against the invading pathogens. In the image, you can see on the left side a normal individual with a normal immune response having the T cells and B cells functional and can fight against infections. Whereas on the right side image, you can see a SCID patient who has a deranged immune system and the T cells and B cells are not functioning properly, so there is a decreased immune response and it cannot fight against infection. What are the symptoms of SCID? Ear, nose and throat symptoms. Infections due to SCID can cause ear, nose and throat problems like congestion, airway infection, thrush, oral ulcers and ear infections. Lung symptoms. Infections like pneumonia due to SCID can cause breathing problems. These problems can lead to death. Digestive symptoms. Infections due to SCID can cause problems with the baby's digestive system like vomiting and diarrhea. Without treatment, these problems can lead to hospitalization or death. Whole body symptoms. SCID can also cause other problems. Babies with SCID may grow or gain weight slowly. Some types of SCID can cause thinking problems, hearing loss and bone problems. The right approach for SCID treatment would be to give the patient a functioning ADA which breaks down the toxic biological products. In some children, ADA deficiency could be cured by bone marrow transplantation where defective immune cells could be replaced with healthy immune cells from a donor. In some patients, it can be treated by enzyme replacement therapy in which functional ADA is injected into the patient. During gene therapy, the lymphocytes from the blood of the patient are removed and grown in a nutrient culture medium and given back to the patient. Here in this image, you can see the different types of therapy for ADA or SCID patient where HLA identical bone marrow transplantation is done, which is the treatment of choice. Alternatively, MUD or haploidentical BMT can be done. Number 2, you can see the enzyme replacement therapy where weekly injections are given with PEG-ADA. You can see the third one where retroviral hematopoietic stem cell gene therapy can be given with reduced conditioning. All these have an effect on the immune reconstitution where the hematopoietic stem cells are stimulated, pluripotent stem cells are stimulated, and slowly all the B cells, T cells and NK cells that are formed are very efficient. It also helps in reduction of hypothyroidism, in diabetes mellitus. It also produces anti-ADA antibodies which can destroy the ADA that is surviving in the body. And it also helps in reducing thrombocytopenia and also in hemolytic anemia reduction. What is done in gene therapy? A healthy and functional human gene, ADA complement DNA, encoding this enzyme is introduced into the lymphocytes using a retrovirus. The genetically engineered lymphocytes are subsequently returned to the patient. Since these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. The disease could be cured permanently if the gene for the ADA is isolated from the bone marrow cells and are introduced into the cells of the early embryonic stages. Here you can see the process of gene therapy in SCID. SCID means severe combined immunodeficiency. ADA means adenosine deaminase. Here the bacterium carrying the plasmid with cloned normal human ADA gene is taken, and the genetically disabled retrovirus is taken as a vector, and the cloned ADA gene is incorporated into the virus, and the retrovirus infects the T cells and transfers the ADA gene into the cells. T cells with disabled ADA gene is isolated from the SCID patient, and these cells form the ADA, and these are grown in culture to ensure ADA gene is active, and genetically altered cells are re-implanted to produce ADA in the child. Now let us move on to the stem cell therapy. Stem cells are undifferentiated cells found in most of the multicellular animals. These cells maintain their undifferentiated state even after undergoing numerous mitotic divisions. Stem cell research has the potential to revolutionize the future of medicine with the ability to regenerate damaged and diseased organs. Stem cells are capable of self-renewal and exhibit cellular potency. Stem cells can differentiate into all types of cells that are derived from any of the three germ layers: ectoderm, endoderm, and mesoderm. Here you can see the embryonic stem cells, where the stem cells multiply and form more stem cells. And these stem cells can be differentiated into different types of tissues, like the adipose cells, muscle cells, red blood cells, neurons, epithelial cells, etc. In mammals, there are two main types of stem cells: embryonic stem cells called the ES cells and the adult stem cells. In the right side image, you can see the embryonic stem cells which are present in the inner cell mass, and these embryonic stem cells later develop to the whole individual, and the inner cell mass is the source of the embryonic stem cells. In the adult, the stem cells can be found in the adult RBCs, WBCs, platelets which are formed in the bone marrow. Embryonic stem cells are pluripotent and can produce the three primary germ layers: ectoderm, mesoderm, and endoderm. Embryonic stem cells are multipotent stem cells that can differentiate into a number of types of different type of cells. Here you can see the fusion of the sperm and the oocyte results in the formation of the zygote, which get transformed into a morula which has a totipotent stem cells. These totipotent stem cells of the morula get differentiated into a blastocyst and forms the inner cell mass, which is the source of the pluripotent cells. These pluripotent cells can differentiate themselves into the circulatory system cells, the nervous system cells, the immune system cells, etc., which are all the unipotent cells. Embryonic stem cells are isolated from the epiblast tissue of the inner cell mass of a blastocyst. When stimulated, the embryonic stem cells can develop into more than 200 cell types of the adult body. Embryonic stem cells are immortal. They can proliferate in the sterile culture medium and maintain their undifferentiated state. Here you can see the fertilized egg, which forms the four-celled embryo, and slowly the morula, and slowly develops into a blastocyst and forms the blastocele. The inner cell mass of the blastocele, the cells are harvested from them and they are cultured in the in vitro and forms the embryonic stem cells. Now, adult stem cells. Adult stem cells are found in various tissues of children as well as adults. An adult stem cell or somatic system cell can divide and create another cell similar to it. Most of the adult stem cells are multipotent and can act as a repair system of the body, replenishing the adult tissues. Here you can see the location of various adult stem cells in the dental pulp, in the synovium, in the nervous tissue, in the adipose tissue, in the cartilage, the hair follicle, the heart, the skeletal muscle, the pancreas, the gastrointestinal tract, and the skin. The red bone marrow as a rich source of adult stem cells. The most important and potential application of human stem cells is the generation of cells and tissues that could be used for cell-based therapies. Human stem cells could be used to test new drugs. Here you can see the potential uses of stem cells. The stem cells can be used in the central nervous system for stroke, traumatic brain injury, learning defects, Alzheimer's disease, Parkinson's disease, for missing teeth, for wound healing, for bone marrow transplantation, which is currently established, the spinal cord injury, osteoarthritis, rheumatoid arthritis, baldness, blindness, deafness, amyotrophic lateral sclerosis, in myocardial infarction, muscular dystrophy, diabetes mellitus, and multiple sites of cancers, and in Crohn's disease. Here you can see the usage of stem cells in the drug development, where the patient cells is taken and these are programmed and the pluripotent stem cells are cultured, which can be helpful in producing different types of unipotent cells like the cardiac cells, the liver cells, and the neural cells. An in vitro model of the disease is made and the drug trial is done to see the effectiveness of the drug on these cells, and drug and the genetic screening toxicology testing is done, and the drug development is done for the required tissues. What is totipotency? Toti means total, is the ability of the single cell to divide and produce all of the differentiated cells in an organism. What is pluripotency? Pluri means several, refers to a stem cell that has the potential to differentiate into any of the three germ layers like the ectoderm, endoderm, and the mesoderm. Multipotency, multi means many, refers to the stem cells that can differentiate into various types of cells that are related. For example, blood stem cells can differentiate into lymphocytes, monocytes, neutrophils, etc. What is oligopotency? Oligo means few, refers to the stem cells that can differentiate into few cell types. For example, lymphoid or myeloid stem cells can differentiate into B and T cells but not the RBC. What is unipotency? Uni means single, refers to the ability of the stem cells to differentiate into only one type of cell. Now in this image you can see the totipotency, the pluripotency, the multipotency, oligopotency, and unipotency. A fertilized egg which develops into a morula has the totipotent stem cells, and this gets transformed into a blastula and a blastocyst. The blastocyst, which has the inner cell mass, has the pluripotent stem cells, which are isolated and cultured in vitro, which can produce pluripotent stem cells. The pluripotent stem cells can also differentiate into hemopoietic stem cells, neural stem cells, and mesenchymal stem cells, which are tissue specific. The hemopoietic stem cells forms the blood series, the WBCs and the RBCs. The neural cells form the cells of the nervous system, and the mesenchymal cells form the connective tissue, cartilage, fat tissue, bones, etc. Now let us move on to stem cell banks. What are stem cell banks? Stem cell banking is the extraction, processing, and storage of the stem cells so that they may be used for treatment in the future when required. In the right side image, you can see the stem cell bank with the different gadgets. Amniotic cell bank is a facility that stores the stem cells derived from the amniotic fluid for future use. Stem cells are stored in banks specifically for use by the individual from whom such cells have been collected, and the banking costs are paid. Here you can see the amniotic fluid stem cells, where the collection of the amniotic fluid is done, and this amniotic fluid cells are separated, isolated, and cultured, and the human amniotic fluid stem cells can be used in treatment in vivo. Cord blood banking is the extraction of the stem cells from the umbilical cord during childbirth. The baby is born with the umbilical cord attached. The cord is clamped and cut so that the baby can be cleaned and taken care of. And the blood is collected from the umbilical cord and analyzed. The cord blood will be drawn from the clamped cord into a special collection bag by the doctor. The blood is sent to a laboratory for analysis, and the red blood cells are separated. Storage. The cord blood stem cells will be kept in liquid nitrogen storage tanks at -190 degrees Celsius inside a secured facility. While the umbilical cord and the cord blood are the most popular sources of stem cells, the placenta, amniotic sac, and amniotic fluid are also rich sources in terms of both quantity and quality. What are the sources of stem cells from the placenta? The human placenta, the placental extracts. The cord blood cells also produce placental multipotent stem cells. The amniotic membrane, chorionic membrane also gives stem cells. The placental tissue fragments also produce stem cells. So today in applications of biotechnology part 2, we discussed about the gene therapy, stem cell therapy and stem cell banks. So kindly subscribe, like, share and comment our channel Read MedPrep Academy. Register for UG and PG NEET type MCQs in our website www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read MedPrep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. To join Read MedPrep Academy, WhatsApp the number given below. Thank you very much.